Welcome to Applied Photophysics, second installment of our webinar series, Beyond Alpha Helix and Beta Sheet, the expanding role of circular dichroism. I'm your host, Keith Solomon, and today's webinar is being given by Dr. John Gabrielson from KBI Biopharma. After a few brief introductory remarks, I'll introduce our esteemed guests and begin the main portion of our program. Before we get started, I'd like to remind all our viewers that our program today will last for approximately 45 minutes, with 5 minutes of introductory remarks, a 30-minute presentation by Dr. Gabrielson, which will be followed by 10 minutes of questions and answers. If at any point during the presentation you have a query, please submit your question in the provided window. We will be reviewing the questions as they are submitted and will choose the best, most germane questions to pose to Dr. Gabrielson at the end of his presentation. Also, there'll be a very short survey after the webinar has finished, which will pop up automatically for all attendees. So please take a few minutes to complete the survey in order to help us improve the webinar series. First, I'll begin by introducing the sponsor of today's webinar, Applied Photophysics. Applied Photophysics manufactures CD spectrometers that allow for the precise, well-resolved, and reproducible analyses of complex biomolecules. Consequently, the spectra produced by the Chirascan technology platform is quite amenable to mathematical modeling and statistical testing, which in turn allows for objective go and no-go decisions and supports the publication of data with true confidence. For more information concerning the Chirascan technology platform and the Applied Photophysics line of stop-flow instruments, please visit us at www.photophysics.com. Now on to the main event. Dr. John Gabrielson is currently Vice President of Analytical Sciences at KBI Biopharma, where he oversees the Elion Labs division in Louisville, Colorado. John joined KBI when Elion was acquired in early 2018. Prior to joining KBI, John co-founded Elion Labs in 2015, where he served as President and CEO and was accountable for defining Elion's strategic direction and implementing its business plan. Prior to founding Elion, John led a multidisciplinary analytical organization at Amgen focused on developing and implementing biophysical and particle characterization methods for higher order structural characterization and comparability testing of all biologics from late-stage clinical development through commercialization and post-approval. He has developed improvements to biophysical characterization methods from which he has delivered invited presentations, been issued a patent, and written numerous articles and book chapters. John received his Ph.D. in chemical engineering from the University of Colorado. His research interests include protein higher order structure characterization, aggregation, and particle formation mechanisms, as well as application of statistical modeling and simulation to evaluate analytical methods. So without further ado, Dr. John Gabrielson will present using circular dichroism to quantitatively compare the higher order structure of protein biologics. Dr. Gabrielson.
Thank you, Keith, for the kind introduction. And as I begin, I want to start by thanking Applied Photophysics for the opportunity to present this webinar today. In this webinar, I'll be discussing the use of circular dichroism for a variety of purposes during biologics development, specifically focusing on using CD for quantitative comparisons of spectra. As I'm sure many in this audience are familiar with, proteins are very complex macromolecules that form multiple levels of structure. And as you'll see on this background slide, proteins form a hierarchy of structural levels, beginning with primary structure, the amino acid sequence, and building into secondary and tertiary structure, which I will refer to in this presentation as higher order structure, and in some cases, subunits combining to form quaternary structure. Just as there are a number of different structural levels that proteins take, there are a number of different methods that we use to interrogate those structural levels. From an analytical control strategy perspective, in my opinion, higher order structures of protein products are critical product quality attributes. As we know from basic biochemistry, the structure of proteins impacts their activity, and therefore loss of higher order structure will impact the efficacy, at least in most cases, and it's reasonable to assume in many cases that loss of higher order structure may also impact the safety of protein products. So given the importance of higher order structure for biological products, we need methods to ensure that the structure remains in place. We do this in a number of ways. As an indirect measure, we use biological assays that reflect the mechanism of action of the product, and also chromatographic purity assays via their retention behavior also demonstrate that the higher order structure of protein products remains in place. There are a number of characterization tools that we also use, including differential scanning calorimetry and analytical ultracentrifugation, that can help to demonstrate protein higher order structure. In this webinar, I will be focusing on circular dichroism, which is a solution state measure, a more direct way for us to interrogate both the secondary and tertiary structure of proteins. Keith shared a slide similar to this a few minutes ago, demonstrating the Chirascan platform. In this webinar, most of the data that I will be presenting was obtained on a Chirascan Q100. We found this instrument to be a very powerful tool for collecting data both for protein secondary structure in the far UV and also protein tertiary structure in the near UV. In my experience, I've seen circular dichroism used for a number of different purposes during development of biologics, and I've listed some of them on this slide. Elucidation of structure for regulatory applications, INDs, IND amendments, and BLAs. During formulation development and stability. It's a good tool to use while manufacturing processes are being developed and characterized. And for force degradation purposes, CD is a very useful tool to demonstrate both the degradation products that are formed, as well as the degradation rates during force degradation studies. In this webinar, I'll be focusing on the last two listed here, comparability evaluations and analytical similarity studies, where the goal of such studies is to demonstrate likeness between spectra of one material to another. Much of the content in this presentation is taken from a stimuli article that was proposed by Martin and colleagues to the United States Pharmacopoeia. It's a proposed new USP General chapter called the Analytical Procedure Lifecycle 1220. And I've taken figure 1 shown on this slide from that stimuli article. Figure 1 shows the analytical procedure lifecycle through a series of stages, beginning with something called the analytical target profile, which I'll discuss on the next slide, and then moving into the life cycle stages 1, 2, and 3. Stage 1 is called procedure design and development, where we learn about the method and we demonstrate or we define the requirements for the method. Stage 2, procedure performance qualification, which I think most of us are more familiar with thinking of this as method validation. This is essentially the final exam for the method where we demonstrate that it's suitable for its purpose. And then finally, stage 3, called continued procedure performance verification or method monitoring. I'll be discussing how we can take the circular dichroism procedure through each of these stages and actually beginning with setting an analytical target profile. As I discuss the life cycle management approach for circular dichroism, there are a few key takeaways from this presentation. I'll mention them up front here, and then I'll revisit each of these key takeaways as we go through the presentation. First, it's important that we define how the circular dichroism procedure will be used. As I stated before, my focus today will be on using circular dichroism for similarity assessments, where the goal is to demonstrate whether or not two materials are similar to each other. It can be used for other purposes. The important point is that we need to be clear at the outset what the purpose of the method is. The second key point is around data quality, and I have a few slides demonstrating the importance of data quality, which can be reduced in many cases to achieving high signal to noise. I'll spend some time discussing the importance of having a numerical approach for spectral similarity rather than relying on visual assessment, which has traditionally been the approach that's used for spectroscopic methods. And finally, the goal of going through an analytical procedure lifecycle management process is to demonstrate that the procedure is fit for its intended purpose. And so I'll be discussing how we do that. Okay, so establishing an analytical target profile. I think the first question here that many of you have is, what is an analytical target profile? You can think of it as a statement that describes what an analytical procedure must do to be suitable for its intended purpose. There are a lot of various elements that a good ATP must have. I've listed 3 questions here. The first is, what level of data quality is required? And we can be very specific here in terms of exactly what signal to noise level is necessary. What is a meaningful quantitative response variable or reportable result that can be obtained with the procedure? And this is where we need to ensure that whatever method we're using can be reduced to a numerical output. And then third, what factors impact the performance of the analytical procedure? And this is where we need to study the impact of factors such as day and analyst and other types of robustness and intermediate precision factors. If you talk with a statistician about setting an analytical target profile, there's also more of a statistical statement that can be made. And so statistically speaking, an ATP is used to define something called a target measurement uncertainty, or a TMU. The TMU is the greatest allowable imprecision that the procedure may have and still be fit for its purpose. So we can think of this as upfront defining what level of imprecision the procedure can have, and then our goal is to show that the procedure in fact has precision within those bounds. A quick point on when ATP is useful. I'm sure some of you have used an ATP in the past and are familiar with this concept for release and stability procedures. We find it valuable to use for characterization methods as well. Circular dichroism is traditionally used as a characterization tool, and we believe can be used also in a release and stability context. I've included an example ATP at the bottom of this slide. Again, this can be varied quite a bit from what's listed here, but for purposes of this presentation, I feel that this is a good representation of what an analytical target profile can be. So the near UV circular dichroism procedure will be used to confirm preservation of tertiary structure in analytical similarity evaluations. What's important about that first sentence is we state exactly what the purpose of the method is. We go on to say, for this purpose, the procedure must be able to quantitatively determine the similarity between 2 or more spectra with sufficiently low misclassification rates. What do I mean by that? First of all, when the tertiary structure of the protein remains unchanged, the probability of an erroneous result indicating that the spectra differ must be no greater than 10%. And number 2, when the tertiary structure of the protein differs, the probability of an erroneous result indicating that the spectra are similar must be no greater than 5%. So you can see here that there are 2 possible types of errors that we can make: saying that spectra are similar when they're in fact different, and saying that they differ when they're in fact similar. In the first case, where the spectra are in fact similar and we say they're different, that's something that's known as sponsor's risk. And it's important that the sponsor's risk be held to a higher level, or I probably should say it's more important that the second case, the patient's risk, be held to a lower percentage than the sponsor's risk. So the actual percentages here of 10 and 5 can change, but we want to ensure that we're controlling the risk to the patient more tightly than we're controlling the risk to the sponsor. I'll move from here to talk about certain design aspects that are important through the first stage of circular dichroism procedure life cycle management. And I'll go through these next few slides fairly quickly, but this first one is related to features of the instrument that as analysts we really have little control over. The Chirascan Q100 uses an avalanche photodiode, a solid state detector, to improve the quantum efficiency significantly. And secondly, efficient lamp coupling and the double polarizing design of the monochromator gives the best output and purity, we feel. And I've compared a number of different CD instruments and find that the instrumentation features in the Chirascan Q100 do outperform other instruments on the market. Secondly, we need to calibrate the instrument. Traditionally, calibration has been done using a chemical calibrant, something like CSA, shown chemically at the bottom here. The issue with using a chemical calibrant is in many cases these are unstable, they may be photolabile, hygroscopic, and they may have a short lifetime. There's also the issue that when we use a chemical calibrant, we're typically using a single wavelength calibration, 290.5 nanometers. What we can do using the Chirascan Q100 is an optics-based calibration. This eliminates the uncertainty and variability that we see with chemical calibration, and importantly, it also uses a multi-wavelength calibration, so we are able not to assume anymore that there's a linear response at all of the wavelengths like we do with conventional calibration. All right, now we move on to a couple of features of the instrument that we as analysts have the ability to control. One of those is the concentration of the sample that we use, and another is the scan rate. And I've included some examples here that I received from applied photophysics where they've measured the tertiary structure of lysozyme in the near UV and demonstrated as a function both of the concentration of the sample and as a function of scan rate how the signal to noise changes. A couple of really important points here. First of all, the fact that the instrument does provide such high sensitivity facilitates analysis when time is limited or when the sample quantity or concentration is limited or when the sample is photolabile. So we have the opportunity to vary scan rate, for example, and maybe give in a little bit on signal to noise for the sake of being able to scan the sample more quickly. Or if we're presented with a situation where we have a low concentration sample, we can still analyze that successfully. Going back to designing the method though, we should be clear at the outset of the method design process what the necessary concentration is for our samples to achieve the level of signal to noise necessary to ultimately get the precision that we need the method to have. All right. Moving on to the second major area within the design space, and that is defining the results of the method quantitatively instead of visually or qualitatively. And I think in the past, for decades, CD has been used mostly as a qualitative test where a subject matter expert views the spectra and determines whether the spectra are in fact similar. And I would refer everyone to the example that I've included here. You can find the full example in the Analytical Biochemistry article. I'll walk through it quickly. And for the purposes of what I'll be discussing, you can disregard the blue spectra on this slide and focus on only the black and the red. This is an example on the left of some FTIR spectra and on the right of near UV CD spectra. In the case of FTIR, I think most of us, when we look at the black population of spectra compared to the red, we would say that those look visually similar. And in the case on the right, when we compare the black to the red, I think it's much more debatable. And some of us may say, yes, those appear comparable. We might say some of the fine structure looks a little bit different between the two. So we may come to a conclusion that they're not similar. And in both cases, we would be wrong. And that's the risk of visual assessment. So in the case of FTIR, we said visually, we would consider this to be a pass. By numerical assessment, it would fail. And in fact, the red is from a structurally altered sample. And if you look at the population of measuring the red spectrum multiple times in the black population, more than 10 standard deviations apart. By any number of numerical assessment approaches, we can demonstrate that these are in fact significantly different from each other. The opposite happens in the case of the near UV spectra, where the numerical assessment actually says, well, you know, these two are both taken from native-like spectra and they're within 3 standard deviations of each other. So in the case of the FTIR, that's a patient risk. In the case of near UV CD, that's sponsor risk. Importantly, in both cases, the sponsor and patient incur greater risk with a visual evaluation of similarity because of the subjective nature of the assessment. Okay, and that leads me to discussing a few different approaches that are available for comparing spectra. The one that I'll focus on is something called the weighted spectral difference, which is an approach that we developed some years ago that uses a weighting function to emphasize regions of the spectra with greater signals. So we're focusing and emphasizing on those regions that we think are more. But there are other methods that are available for comparing spectra quantitatively, including the correlation coefficient, the area of spectral overlap, the derivative correlation algorithm. I've seen a publication related to an adaptive multivariate curve resolution approach, Euclidean distance, and as I said, the weighted spectral difference. As we consider what type of numerical approach is best suited to the type of experiment we're running, it's important to think about what types of spectral differences are important. Is it the absolute difference between two spectra that we're most interested in? Is it differences in their shapes? Is it the local details, the fine structure? What we kind of settled on when we were going through this assessment was the fact that not all parts of the spectra contain equal information content. And therefore, we felt that the weighted spectral difference, because it could handle situations with different signals, it would apply different weighting. We felt that that was a good approach. Okay. I have a few comments related to stage 2 validation, and then one quick slide at the end related to monitoring. So now that we've sort of designed the method to a certain set of requirements that we need, including achieving the signal to noise that we need, reducing the output to, using a quantitative response that we think matches the type of analysis that we will be performing. At this stage, we move on to the validation of the method. And there are a couple important points to think about during method validation. We need to understand how the method behaves under actual conditions of use. So when we look at multiple pieces of equipment, multiple analysts across a time period, we need to assess things like the linearity, range, and specificity of the method. And most importantly, its precision. It turns out that the precision of the method is the most important performance characteristic of the method. We also need to study the method under more extreme conditions. And this can be more moderate, like process excursions, things like temperature, pH, light, hold time, things that the material may actually undergo during routine processing. And it can be also under forced degradation conditions outside of what we would think the protein product would undergo during routine processing. The reason that we perform forced degradation experiments is to deliberately and irreversibly alter the higher order structure of the protein to ensure that when the structure actually changes, that the method is sensitive to those changes. And then as we go through this process, we ask the question, does the method perform adequately to achieve its purpose? As I said earlier, I will be focusing on, or am focusing on, comparability and biosimilarity applications, where the purpose of the method is to assess the likeness or the similarity of one type of material to another. To achieve this particular purpose, the method must be able to discriminate between materials for which a meaningful difference in the measured attribute actually exists. We have to show that when there is actually a change, the method can detect that change. This is important because we must be able to distinguish between lack of method sensitivity and lack of change in a measured attribute. If we were to measure 6 samples and they all look the same without intentionally altering any of them, then we don't know if the method doesn't see a change simply because it's insensitive to change or because we didn't induce a change. All right, so we can think of this method validation exercise as one that would kind of walk through these 3 steps that I've highlighted on this slide. First, defining the quantitative response, which I've already spoken to during the design stage. Secondly, calculating the precision and defining something that I call a detection threshold. And third, modeling the data and calculating the sensitivity of the method. So we've already covered that first one around defining a quantitative response. So I'll focus on the second and third of these. We've found over a significant amount of time with a number of different proteins that the precision of the circular dichroism method, and I should say both in the near UV wavelength region and in the far UV, tends to be consistent across proteins. Note that I'm not saying that the spectra themselves look the same for different products, but the precision of the method when you study different manufacturing lots of the same protein and even across different proteins tends to be similar. This gives us the opportunity to combine the precision, or to sort of combine the data we've collected across a large number of proteins together into a single estimate of precision. Once we've done that, that gives us a way in which we can estimate something called the detection threshold. We can then run an experiment where we intentionally alter the protein in some way and calculate the point at which we can detect that change when it crosses the detection threshold. And I'll go through an example of doing that in a couple of slides. Before doing that, I wanted to spend just a minute discussing in a little more detail this idea of universal qualification, which simply means that we can define estimates of method performance across a number of different proteins, even across classes of proteins. And this is important because, first of all, we think it's a more robust measure of method performance because it includes a lot more data. And secondly, it's a much more efficient way to validate the method because you're not having to revalidate the method for every product. This is just an example, the graph, showing a variety of different measurements. It's a total of about 300 spectra measured over about 3 years, 10 different proteins. And what's important to note here is you do see some fluctuations in the weighted spectral difference is essentially here, in this case, a measure of the precision of the method. And you can see that it does fluctuate across that time period, but that has more to do with the date of analysis than it does the product. And you can see that because in some cases, the same product measured 6 months or a year apart has very different levels of weighted spectral difference. So what we were able to do is take this, again, very large body of data and partition the variance into sources. So we looked at a variety of different sources of variation. We had the metadata that we needed to do this and parsed it out into residual date, product, lot buffer, and analyst related variance. And as you can see, the majority of the variance, in fact, just over 50% was from random error, residual variance. And then about a third of it was from day-to-day fluctuations in instrument performance. All of the other terms that we looked at were insignificant compared to the residual and the date variance. So again, once we have this kind of knowledge, we can build that detection threshold and use it across multiple products. Okay. The last step within what I'm considering, at least for our purposes today, as stage 2 is to demonstrate the sensitivity of the method. And we do that, as I stated before, by intentionally degrading the protein in some way. And this can be done through thermal degradation, through chemical means, or even very targeted degradation. We've, for example, used oxidation as a means to chemically change the protein from which we believe there's a change in the higher order structure that we can measure by CD. Regardless of the means, we want to induce spectral changes, as I've shown here, and then across a range of that, either impurity or thermal denaturation or chemical denaturation, measure a number of different points. We can fit a line through that and then determine the point at which it intersects with the detection threshold. This is a measure of the sensitivity of the method. Before I leave this slide, very quickly, I wanted to focus or go back to the target measurement uncertainty. As a reminder, the target measurement uncertainty is what we said at the beginning was what the method needed to be able to do, the allowable amount of uncertainty in the method that would still allow it to be suitable for its purpose. Once we've gone through the validation stage, we have the data that we need to demonstrate that this detection threshold that we're showing here is in fact below the target measurement uncertainty. And it's not quite as simple as that. And I would ask all of you, if you want to go through this exercise formally, to work with a statistician on it, because you should look at the upper 95% confidence bound on the estimate of the precision, not the actual estimate of the precision itself. But in essence, what you're doing is you're demonstrating that the actual precision of the method is in fact below the target measurement uncertainty. All right, one quick slide on monitoring, and then I'll summarize at the end. So the final stage of lifecycle management is ongoing monitoring of the method. And this is a step that is often not performed for characterization. One, I believe, that should be. We need to demonstrate on an ongoing basis that the procedure remains in a state of control. And one way to do that is through this sample sequence that I've included on the right-hand side of the slide, where every time we run the CD procedure, we have an in-house reference standard that we use as the first measurement we make, followed by a series of samples up to 10. And then we bracket at the end with a second in-house reference standard. What we do then is we take the ending standard compared back to the initial standard and calculate the weighted spectral difference between those two. If that result falls within a certain boundary, below an upper threshold, then we're able to accept the results of all the samples in between. And I think that's fairly common. I mean, we do something similar to that with chromatographic purity methods all the time. What we also do is we take that first reference standard and compare it back to a primary standard that we measured on a different day. And the reason we do that is to control long time drift of the method. And then, so a cross day drift. The end to beginning on the same day is to control within day drift. And on the bottom, I've shown an example of a trend chart that can be used. And this is actually not real data. This is just sort of simulated data showing, initially the method was in a good state of control. We've been monitoring every day that we use the method. And then over time, it looks like maybe it's drifting outside our control point. And so that's why it's important to trend the data so we can go back, kick back to redesign of the method at that point. All right, and with that, I'll summarize a few of the key points from the webinar. First of all, I hope I was able to describe why visual assessment of spectra is insufficient. And I don't mean to say that we shouldn't look at the spectra. Of course we should. But when it comes to a determination of similarity, it's important that we also have a way of reducing that spectral output into a number that demonstrates the degree of similarity between those two spectra. Secondly, it is possible to manage the circular dichroism procedure and other characterization methods for that matter through 3 life cycle stages to demonstrate its fitness for purpose. The design phase, when we really try to understand the method, what it can do, and define the requirements for the method. The validation stage, where we demonstrate that the method is suitable for its purpose, and finally, ongoing monitoring of the method. And then finally, a life cycle managed circular dichroism procedure can be used confidently to characterize protein higher order structure for many different applications within biologics development, including what I've focused on, which is analytical similarity studies. With that, I'd like to thank my colleagues from Applied Photophysics, Keith Solomon and Luke Hitchcock-McCarthy, for their help, both with the content of this presentation and with setting up the webinar. And my colleagues from Elion Labs, Brent Kendrick, Kelly Arthur, and Rick Burdick, who helped immensely with helping to put the content together and the ideas behind a lot of what I shared today. And with that, I'd like to thank everyone for their time, and I'm happy to take any questions.
Thank you, Dr. Gabrielson. I believe our viewers really got a good lesson on how to model CD data. And we do have some questions from some of our viewers. First, I'd like to thank everyone for their attention. But here are some questions that people have submitted during the webinar broadcast. One viewer writes in, my students and I use CD to measure the secondary structure of peptides and membranes. We use a thin film method to embed the peptides and lipids first, then completely evacuate the solvent and then hydrate through the vapor. We tried using usual algorithms in Dichroism, but they did not work. We used the shape analysis method of Norma Greenfield and coworkers. Are you familiar with this least squares fitting method?
I think, so thank you, I appreciate the question. I think the short answer to that is no, I'm not familiar with that specific method. I can understand the difficulty from the question of what you're trying to accomplish. We have found in our experience looking in the far UV range that there are a number of different tools that can be used to estimate the amount of different types of secondary structure, to sort of deconvolute the secondary structure. And to be honest, I've not seen a lot of value and certainly not a lot of reproducibility using those tools. So I think this is one that I'd be happy to kind of exchange some emails around. If I can get your contact information, I can discuss certain methods that we've looked at for doing, I think, similar work to what you're doing. But I'm not familiar with the specific approach that you're using.
Thank you very much. Dr. Gabrielson, another viewer writes in, can you explain the variance observed from the dates of analysis?
Yeah, that's a very good question. It's something we've spent some time trying to think about. We do know that over time the instrument changes. We don't know exactly what's happening, if it's the optics, the mirrors, but upon after the instrument has a PM, we do believe that the signal to noise does improve and the data quality does improve, and you see that via the reduction in weighted spectral difference results for one standard, for the same standard compared to itself. And so what we tend to do is measure our what we call primary standard immediately after the PM of the instrument, so that we can have more like a gold standard to compare back to, so we can monitor that degree of drift. The graph that I think you're referring to, that was intentionally chosen where we found a time period that showed about the most dramatic change that we've seen. So it was meant to highlight the point that there can be day-to-day fluctuations. I would say what you saw there is a bit on the more extreme side of what we're used to.
Thank you very much. Another viewer writes in, does the instrument record the in-house reference every time? If yes, how do we assess this reference? Also, does the instrument negate the in-house reference every time we subject a spectra for analysis?
Let me start by saying I'm not sure that I was clear on what I meant by the in-house standard. This is something that Elion Labs has that we use as our own standard. It's like another sample that we're running. So the instrument is just performing like it would on any other sample, and we are then taking that data and putting it ourselves into a trend chart that we're using over time to evaluate the performance and drift of the method. So I guess I was confused in the question about the instrument side of this, because the instrument really doesn't know the difference between the sample and the in-house standard that we're using.
Thank you. I think that clarifies that important point. Another viewer writes in, how important is the choice of using WSD compared to other analyses? Do you ultimately get a different result or conclusion by using different ways to reduce the spectra?
Yeah, that's a very, very good question. Thank you. And I've spent a very long time looking into that. I would encourage you to look at a couple of papers that I cited in here. The first is the Tesca article, and the second is DINH, D-I-N-H. If you have an opportunity to look up those two papers, I think it'll do a better job explaining this than I can do in the next couple of minutes. But the answer is yes, you can get a different conclusion based on the approach that you use. Now, I would say it's far more important that you do some type of quantitative analysis than to do nothing at all. So in other words, any type of numerical assessment of spectral similarity, I believe, is better than relying on a subjective visual assessment. With that said, there are pros and cons of each of these different tools, and we've seen in some cases where one tool would say that two spectra fall within an acceptable difference, whereas another tool would say they fall outside that acceptable difference, and so you would come to a different conclusion. Again, I think that you have to understand how you're using circular dichroism and the types of studies that you'll be conducting to help determine what type of quantitative tool to use.
Thank you very much. Another viewer writes in, do you mean that CD spectroscopy is unique among other spectroscopic ways of protein analyses? We commonly apply such procedures to HPLC-based chemical analyses.
So thank you again for that question, and I'm glad I have the opportunity to clarify. No, I do not mean that CD is unique in this context. In fact, much of the presentation that I went through today would be equally applicable to other types of methods, and not just spectroscopic methods, but chromatographic methods and other characterization methods as well. In fact, the weighted spectral difference and those numerical approaches to compare spectra can be used just as well to compare DSC thermograms, to compare chromatograms. Well, actually, in that case, modifications do need to be made to use those tools, but in essence, they can be used for those purposes as well. So my point was to show that this is all possible for the circular dichroism method, but that is not to say that these same things shouldn't be also applied to other methods.
Thank you, Dr. Gabrielson. One last question, and this one comes from me. Can you comment about the use of spectral difference versus weighted spectral difference? I think our audience would get a lot of insight over the why weighted spectral difference is an improvement.
Yeah, thank you for the question. So when we were going through the process of defining what we felt was a good tool to use to, again, reduce that spectral complexity into a number, we started with something called Euclidean distance and modified it from there to something called spectral difference, and then eventually to where we are now, which is weighted spectral difference. So really, first, I think I'll take just a minute to explain what spectral difference is and what weighted spectral difference is before answering why we prefer weighted spectral difference. So spectral difference, what it does is at every single point of each of the two spectra, it takes the difference between the two values, it squares the difference, it sums all of that up and takes the square root, and then divides by the square root of the number of data points. That will achieve a number that measures the difference between those two spectra holistically. What we found in using that method is that it applies the same weight or emphasis at every single wavelength point. And what the weighted spectral difference does that we think is an advantage is it focuses more weight, a higher weighting factor, on regions of the spectra that have higher signals. And by doing that, if, for example, if you have a region of baseline where there are some sort of noise differences in the baseline region, we don't think that's important. And in spectral difference, that would be included and weighted more heavily. Whereas in weighted spectral difference, because those values are close to baseline, they're close to zero, they would be weighted very low, and you wouldn't see an impact on the WSD value. Again, I'd refer you to the DIN article, D-I-N-H, that goes into a lot of detail on this.
Thank you very much. I really believe our viewers got a full and comprehensive understanding of how to model CD data. And if there are any other questions, we can take them and email them to you and have you respond offline at some time in the future. And we really thank you very much for all your time. We want to thank all our attendees. It's great having you view our webinar. Recordings of today's presentation will be sent to all registrants tomorrow. Please join us again for the next installment of this webinar series, which will continue to inform our viewers about recent innovations in the acquisition and analysis of CD data. In the meantime, If you have any questions about the Chirascan instrument platform and our other product lines, please visit us at www.photophysics.com.